So, in our last module we have discussed that how to do cell culture in microfluidic platform with reference to 2D cell culture and 3-dimensional cell culture. And at the same time we have also discussed how to perform the different type of experimental setup, like say different types of fluid flow, microscopy, and then we had gone through it that several experiments by which you can utilize this fluid flow to decipher some fundamental behavior of cells in microfluidic system. So, now we are coming how this microfluidic system could be used for application aspects like say biochemical analysis, say organ on chip. So, in this lecture I shall go through that how that organ on chip is developed on the basis of cell culture principle in microfluidics and to some extent tissue engineering aspects to develop that miniature form of that organ on chip. So, what do you mean by the organ on chip? Organ on chip means a class of micro engineered in vitro tissue models done by using small compartments connected by the microfluidic channels and porous membrane for nutrient feeding, inter and intracellular communication. Now, we are coming what do you mean by that micro compartments? We know that different types of cells comes together makes a tissue. Tissues are organized to organ, then organ system, then whole that living system. Now, in a organ the tissues are arranged 3-dimensionally in a different types of micro environment, and those environments are cross talking through that this permeable mechanism or barrier mechanism. So, permeable mechanisms like say free diffusion of the chemicals, maybe porous membranes are involved, means like say in extracellular matrix or basal membranes, then fluid flow in microfluidic channels. And if we look for the barrier mechanism of the environments, say this is here that cells are attached and this is another environment where cells are not attached, means non adhering situations. Maybe different phases like say air and liquid like say alveolar sacs where that air and cells interface occurs. Then physical barrier like say blood brain barrier where that physical barrier is there, not all molecules will not go. In that way that in a organ different micro environments are created and they are cross talking with each other. So, that is why that micro compartments are described in that way, create unique conditions arising from scaling laws, simultaneous and precisely control multiple chemical and physical culture conditions with a spatial resolution appropriate for cell and tissue culture, and manipulate the chemical and physical communication between environments. What I mean by the environments? This is micro environments inside that organs or tissues. So, what are the features of organ on chips? We are coined as OOCs, means organ on chip OOCs. I shall be coined that OOCs as organ on chips. The OOCs typically mimic a minimal functional unit of living organ, not a whole organ, like say portion of cardiac tissues or kidney tubule. Like in kidney tubule is the unit of that kidney organ, or in cardiac system that cardiac muscle or some bundle of fibers, contractile fibers are unit. And these compartments or tissues will be organized to recreate tissue-tissue interface like alveolar capillary interface or blood brain barriers and expose them to their physiological relevant chemical and mechanical environments like say cyclic force or stretching force or fluid shear, etc. So, that is why for the development of most of that organ on chips, they need some mechanical force, electrical force or optical induction, electrical induction for their maturation, at the same time to exhibit their physiological behavior. Multiple organs could be integrated by linking individual organ on chips through a microfluidic channel or through a porous membranes. And most importantly, this organ on chip is a highly multidisciplinary works. It is an extensive integration between molecular biology, cell biology, organ physiology, cell culture, stem cell expertise, microfluidics, microfabrications, material science, and after all that clinical disease knowledge, means physicians or clinicians should be involved to really develop that organ on chips. So, what are the application of organ on chip, means who are the stakeholders of this organ on chip? First thing is that using this organ on chips, we could have an understanding of the organ physiology and pathophysiology of specially human disease, means disease model could be established in vitro rather than based on animal models. Then reduce, refine and ultimate replacement of substantial number of animal experiments, means these are the 3 R's basically. Generally in the any development of the drugs, we are using animal as a model system, but nowadays ethics are coming very strong way. We cannot use animals for our purposes or drug targeting purpose or drug, means clinical trials. So, for that this human organ on chips is very much essential, and human disease model already I have mentioned, and accelerating the drug development by resolving the discrepancies in drug safety and efficacy observed between animal models, cell culture and clinical studies. Because when any drug is coming to the market, first preclinical data, clinical data, then it is going to several phase clinical phase trials, then it is coming. So, there is a lot of discrepancy between the cell culture data to animal experiment data to clinical phase 1, phase 2, phase 3 clinical data. So, those should be rationalized on the basis of human organ on chip. So, if we think that to develop a organ on chip, what should be the design criteria? First thing is that we have to determine the set of functional characteristics and specific question to be addressed in the organ to be model. As already we have mentioned that organ on chip, basically it is a miniature form of that organ, not a whole organ. So, that is why specific questions should be asked, what we should be looking for from that organ on chip. Next is, what are the readouts required to answer the desired question? Like say if we look for that heart as a organ on chip, basically we shall be looking for that how that myocardial fibers are contracting and generating the force, or how many beats are there, and how that electrical polarization will help for that mechanical contraction, and so on and so forth. Then most importantly, if we have a monitoring system which is online for the long term culture, by which you can monitor that non-invasively, that how that organ on chip behaves with respect of communication, with respect of their cellular physiology, oxygen uptake, pH and so on and so forth. Then manipulation of the microenvironment, passive or dynamic stretch, electrical and optical signals, fluid shear and biochemical and hormonal cues, means both biophysical and biochemical cues which comprises the microenvironment, and integration of the multiple organ on chips by microfluidic network which will mimic that different organ in our system, like say heart, lung, liver, brain, kidney, gut in one platform by connecting through fluidic networking, and we can mimic that human physiology. And organ on chip utilize the advantage of control strategies and multiparametric approaches designed for microfluidic system for predicting the organ level response. So, totally engineering approach should be utilized to harvest that potential of organ on chips. So, this is an example how that online monitoring system is developed. So, like say there are two organs, this is a liver and this is a heart, and they are connected through microfluidic platform, and that online monitoring system is developed for say bioelectrochemical sensing module, then physical and chemical sensing module is automatically means without any distraction that fluids are monitored for their cellular behavior. In that way, we can monitor that whole the system like in our body, how it behaves in every days or weeks after weeks, even months after months. So, next question comes, what are the cell to be used for organ on chip development, and already we have discussed that for 2-dimensional cell culture or 3-dimensional cell culture, different types of cells like say primary cells or cell lines or some cancer cells should be we have already mentioned those cells are used, but in human organ on chip development we need cells of high density and always cells are not available. So, that is why induced pluripotent stem cells are used and which should be triggered to develop different types of cells, and those cells should be used. So, source of cells might be primary cells, cell lines and derivatives of induced pluripotent stem cells. Then how much complexity should be in organ on chips? It may be a simple, it may be a complex, but complexity depends that how much, what questions we are asking for that complexity. So, complexity of individual organ on chips and the need of multi organ chips depends on the problem being studied. Like say example, say if you want to monitor that, say mimic that microfluidic structure for heartbeats, say then micro pump can be used to mimic that pulsatile blood flow. That how that pulsatile flow can be could be means arranged in fluidic platform, that already we have discussed in our earlier lectures. Now, we are coming how that single organs on chips are developed like say heart, lung, hepatic globule, nephron, solid tumors and so on and so forth. So, a minimal functional simplest possible unit of each organ system to create organ on chip environment or so called micro environment, and incorporation of biophysical stimuli like say hydrodynamic, mechanical, electrical and chemical that drive and control the establishment of organ model and its minimally acceptable physiological functionality. So, we shall be discussing some of that models like say first we are coming heart muscle for the organ on chip development. So, what are the design consideration for that heart muscle? Contractility and electrical activity, and what cell type should be used? Here induced pluripotent stem cell derived cardiovascular myocytes are used, and what are the readouts? Beat rate, force, excitation threshold, maximum capture rate and contractility. So, here that minimum functional unit is that a bundle of contractile fibers, or you can tell that myocytes, means that when that is contraction occurs, then it generates force and that force are culminated or it is aggregated to make that heart beat basically, and that contraction is induced by that electrical polarization that could be mimicked by that electrodes, and in vivo system this electrical conduction for the polarization of electrical conduction are done by that pacemaking cells or pacemaking node at that sinoarterial node. So, that is mimicked by using these electrodes by depolarization and polarization, they will induce that stretch fibers which will induce ultimately force generating to that media. So, this is bio-wire, means where the cardiomyocytes are embedded in a hydrogel wire format and that is connected with that means electrical force by which that contraction will be there in the cardiomyocytes and generate that force, and that could be monitored. So, this is an example of that heart muscle organ on chip. This is a very beautiful model for that lung on a chip, because unit of lung is alveolar sac. These are the alveolar sac, means it is air sac surrounded by that basolateral membrane, then endothelial cell, means endothelial network and some extracellular matrix protein. This is the unit of your alveolar sac. What is the function of alveolar sac? That is, there is a exchange of gas, means air, oxygen and interstitial fluid carbon dioxide exchange. So, that is that function due to the cyclic stretching of lung, means when you are breathing and exhalting, then that cyclic stretching occurs. So, mimicking that cyclic structure in the microfluidic platform, it is the unit of your alveolar structures or lung alveoli. So, how this structure is formed? So, in the microfluidic system, this is a membrane, porous membrane, biocompatible porous membrane, it is a stretchable membrane. In the upper side of the membrane, that epithelial cells are cultured, and the bottom side of the membrane, that endothelial cells are cultured. After that confluencing, then that media is replaced by air, and that stretching force is given by using that side chambers vacuum. And when that stretching force is adopted, then cells are differentiated to like say alveolar epithelial cells, and that underneath is your interstitial flow, and which behaves like your lung alveoli, and here that gas exchange between that bottom chamber and upper chamber full of air, that could be monitored, and here that microenvironments is your air cell interface and bottom is cell interface through that membrane. So, here cell lines are used, mainly primary cell lines or some epithelial cells are available nowadays, and here readouts are mainly by microscopically cell imaging and dissolves gas concentration by online monitoring system. So, this is a first system, means first discovered system or established system using that biomimetic principle. Then lot of systems are developed using this membrane and that gaseous exchange using that side arm vacuums like in gut in chip. Liver is an organ that it is a chemical factory where that drugs are metabolized, lot of proteins are synthesized. So, what is the unit of liver? Unit of liver is liver lobule. So, lobular structures, this is an example of say representative example of liver lobules. They are arranged in such a way that this is that central vein and hepatic artery, and these are the cells where that hepatic cells are high density arranged, and that venous and arterial systems giving that blood means nutrient flow and then exchange of that liver nutrients and other things. So, how these things could be mimicked in microfluidic platform? Here cell lines are used, means HepJ2 and sometimes induced pluripotent stem cell derived liver cells are used. So, here is that design of that microfluidic platform. This is the unit of that system, that liver cells are high density impregnated in that middle layer, and that outside there is a medium flow, and these are the microfluidic barriers which mimic that structure of your venous and arterial connection to that hepatocytes. So, that cell density is here near or almost equal to that in vivo system, and here that total medium inlet and medium outlet, and medium is perfusing through that where that hepatocytes presents in high density. So, here the readouts are that how that hepatocytes secretes albumin, urea cytochrome P450 enzyme activity, metabolite conversion and drug induced liver injury. Means with this system some disease model also could be developed. And this is kidney on chip. That kidney function is that glomerular filtration, and it is that here blood is flowing through that channel of that glomerulus nephrons, and these are the cells means where that plasma salts, small proteins, glucose phosphates are in, and that proximal tubule where that uptake is there, then they are means not absorbing urea and some salts, they are passing, ultimately they are excreting as a urine some salts and urea. So, this is the structure of kidney, this is your nephron, unit nephron, and how this could be mimicked, because that epithelial cells in the nephron that are highly polarized. So, for the polarization you have to give that fluid flow. So, with the knowing this principle, so that matrix is developed here, that porous matrix above which that epithelial cells are growing, and below that interstitial fluid means that interstitial flow is flowing. So, this is the tubular flow, means this is the tubular flow of blood, and here is the interstitial fluid. So, if we give this fluid in this microfluidic chamber, then it is observed that epithelial cells are polarized, the apical and basal, and they express their functional means transport proteins. So, in the experiments all types of epithelial cells are generated, and they are used for that to mimic that whole that tubular structures used at that level of proximal, Henle loop and distal sides. So, totally it could be mimicked that filtration structure of that kidney. So, in that way lot of other organs are developed in organ in chip module, like say gut, blood brain barrier, skin, vasculature and cancer models. So, in the gut design consideration the drug absorption for the large surface area via the villi, microvilli formation and mucosal barrier, and symbiotic bacteria means microbiome what is present in our gut. Then cell lines are used, say CACO2 cells are used, and the readouts are transepithelial transport absorption, toxicity, cytochrome P450, 3A4, isoform drug metabolism and response to bacteria. In the same way for blood brain barrier, the readouts are transendothelial residence, permeability and drug transport. For the skin, transdermal transport and gene expression. For vasculature, permeability, response to shear stress and transendothelial resistance. And for tumor, lot of things already we have discussed to some extent in our earlier lecture, and for the tumor model, that metastasis and how that is extravasate from one place to other place. So, after discussing that organ on chips, now we are coming how these organ on chips could be integrated to mimic that different organ system, how they are connected in our body. So, that is integration of multi organ on chips through microfluidic platform. So, here that basic principle is that communication between that organs through soluble factors and extracellular vesicles that mediate peripheral crosstalk with the circulatory system. So, for that microfluidic mimics are used, like say in our vascular system, perfusion system, and enables it control over the culture environment and recapitulate some of the aspects of homeostasis. And these connections like our in vivo system helps to crosstalk, at the same time feedback control mechanisms what is happening in our normal physiology. Now, how these organ on chips will be integrated in different configuration, means maybe parallel, serial, or in one platform, or in a continuous fashion, or in a perfusion readouts or perfusion ways. So, one by one we are coming. that first one is integrating multiple organ chips in a static organ chamber, means static organ chamber means there is no convective flow, in only in one chamber that different organ on chips are there, or in a big say petri dish, that small petri dish are there where that individual organ on chips there and they are bathed in same your media. That is multiple single organ on chips can be cultured within a shallow wells, each containing tissue specific media and then connected by the media within the larger wells. So, this is that first module, means what are the drawbacks of this module is that there is no convective flow what is happening in our body. See, in that way some of the examples are liver fibroblast, here is no perfusion, increase in toxicity when metabolic activity of hepatocytes are present, liver, kidney, lung, neuronal vasculature and cancer, which means at least 4 or 5 organ on chips are together. Then gut liver in that transwell based model contaminates containing the CACO2 cells with transwell and hepatocytes. Then integrating multiple organ on chips by single pass perfusion means their convective flow is there. This is common media inlet and common media outlet, means in that fashion these organs on chip could be connected by either parallelly or in series or in both ways. But problem of this type of configuration is that downstream of that flow there are no feedback, they are controlling only from that upstream, because upstream organ on chips they are not getting the feedback from the downstream. So, examples in that system, that single pass vascularized tumor, hydrostatic pressure is used, heart, diffusive transport, gut and liver, gravity driven unidirectional flow. And this is a chip where that recirculation is there, means like your normal vascular system, that common media is passing through that each of the organ on chips and again circulating. This is like a circulating system in our body. So, in that platform that every organs they are getting feedback from their other organ, there is no downstream or upstream. So, they are getting in that way. Then last module is that where that each organs are connected by that perfusion modules. Here recirculation is there and each organ, means each organ on chip they have individually cultured in their individual media and then they are connected through a means common channel with endothelium and where that immune cells will passing on, and this is the circulating flow direction by which all cells are connected keeping their individuality. So, these are the some of the examples where that perfusion flow are induced for that recirculation purposes, liver, lung, kidney, liver, heart, liver cancer and etc. So, now that utilizing this organ on chip in a connected way and how these systems are used in drug development. If we look for the drug development project, they are passing through different stages. So, first is drug discovery, means where it works, drug discovery means which is the target, then preclinical screening like say in cell culture level or animal model level. If then pass, then it is going through clinical studies, then phase 1, phase 2, phase 3 clinical studies, then it is coming to the market. But real sense, most of the drugs they are not passing after their preclinical stage, because most of the drug development situations are done in cell culture model or in animal model, because there is a huge amount of discrepancy between these and human system. Again after that 2 or first clinical trial or second clinical trial, these are failure due to the different level of populations. So, how this could be addressed using this organ on chip model. So, like say this is an example, say we are looking for a drug for cardiovascular disease first question we are asking, does this drug causes cardiovascular side effects? Means first is target identification, means say in the heart, whether it may be a heartbeat is affecting or blood pressure is affecting. If target is identified properly, then if there is a in the target level there is a any toxicity, means if this drug works on heart muscles contractility or it affects your blood pressure or if that uncontrolled heart beating, means arrhythmia, means this is a side effect in the target level. Then if you look for that side effects in other organ level, means then does this drug causes toxicity to other organ, means this is a cardiovascular drug, whether it affects liver or whether affects the bone marrow, whether affects his brain, means blood brain barrier. So, these intra and inter toxicity, drug toxicity could be assayed in a microfluidic platform using different organ on chips models. Then if that mechanism causing that altered functionality or does this drug work on the intended target, if it is passed through all these things, then possibly we can go for that clinical study. So, if it is not passes through these studies like in organ on chip model, then it should not proceed further, means this will help drug development project to proceed or not to proceed in earlier stage, and it will decrease that cost of that drug development. Even if that it is possible to do that drug trial in patient basis, say, because in most of the cases clinical trial that terminal patients or who are means highly disease situation, they are not included, because due to that they should not be trial with that drug trial. So, most of the drug trials are done by that population level with some normalization, they did not consider the ethnicity genetics of that humans or something like that. So, those should be considered in the personal basis in that organ on chip model. So, organ on chip can provide mechanistic studies on drug actions, preclinical trials, clinical studies using patient specific organ on chips, and most and at the last that clinical trial on chip to discover therapeutic options and for rare diseases. Means organ on chip has a huge application to that in the coming fields, but there are a lot of challenges. What are the challenges? That modeling of drug absorption, distribution, metabolism and excretion, ADME, then scaling of organ size and vascular flow, because it needs a lot of means simulation and engineering aspects to scaling the organ size and vascular flow. Then throughput of that organ on chips. Right now what organ on chips are available, their throughput is not very much high, what pharmaceutical companies want. So, high level or high throughput level organ on chips are should be developed, and last of all, so far there is no organ on chips where that immune system, endocrine system and neurological systems are integrated, because we know that each and every organs that high level neurological controls are theirs. So, those type of integration is necessary to mimic that real organ on chips, which will mimic that in vivo physiology or in vivo system.